Fluorescence recovery after photobleaching. So we know that proteins diffuse throughout the cell membrane. How can we tell how fast proteins move throughout the cell membrane and what affects the speed? We can use fluorescence recovery after photobleaching. Let's say we want to find out the speed of protein A, a membrane protein. First, you want to introduce fluorescence so you can see protein A under a microscope, usually a confocal microscope. You don't want to use an antibody because you want to observe a live moving cell that is still functional. Instead, you can introduce a fusion protein. You can introduce GFP into the genome by recombination, and so whenever your gene A is expressed and made into a protein, you'll also have a GFP attached to that. Usually a downside to fluorescence is photobleaching. If you expose it to light for too long, you'll lose the color. But in Frap, we use that to our advantage. Here's our membrane protein A visualized with GFP. So we'll pick an ROI, or region of interest, on our cell. Then we'll use a high-intensity light or laser on our region of interest. Then you'll observe your ROI over time to see how quickly other protein A moves into your bleached region. From that, you can conclude how mobile your protein is. For example, the arrow represents when the laser is used to bleach your ROI. You can see that fluorescence dips and starts to recover over time. In this case, your level of fluorescence doesn't recover, and you can conclude that your protein is immobile. So how fast does our protein A move? From using Frap, we can tell that protein A is highly mobile. Frap has many applications. You can also use Frap to see how sugar modifications and protein folding affect protein mobility. Frap has also been used to study protein aggregates in the brain to better understand how diseases like Alzheimer's and Parkinson's work. Thanks for watching.